All right, welcome everybody. We're going to talk DNA structure and replication today. You should have your notes ready to go. I have to talk really fast because this video can only be 10 minutes long. So while I'm talking, I want you to pause and take your notes and then come back to me as need be. Okay, and let me know in class if you have any questions or concerns. All right, when we talk DNA structure, we have to start at the 7th grade level just to make sure everybody's in the same place. First of all, what is DNA? Well, DNA stands for deoxyribonucleic acid. You're going to learn later that deoxyribose is the primary sugar. Yes, there's sugar in your DNA, but we'll learn that in just a minute. DNA is found in the nucleus of every cell. It contains the genes for an organism, and it determines what we're going to be like physically and even emotionally. Now, obviously, environment also plays a role in that, but it turns out our DNA has a lot of information both about who we're going to be physically, but also who we're going to be in terms of our personalities. All right, I want to clarify a few things. Sometimes people get the terms DNA, chromosome, and gene mixed up. They're interrelated terms, not too complicated. So what's a chromosome? Chromosomes are just twisted strands of DNA found in the nucleus of every cell. There's a photograph of bivalent chromosomes right in front of us right here. So how are chromosomes, genes, and DNA related? Well, chromosomes are made of DNA, and sections of chromosomes are called genes. You have 46 chromosomes, and on those 46 chromosomes, you have between 20 and 25,000 genes. The rest of the DNA that is not part of a gene is considered junk DNA. Now, that doesn't mean it's not important. It's just not being coded for a protein, and you'll understand that as the unit moves on, as we learn more about what DNA actually does. All right, so quick bit of history. I don't know if you were taught this in 7th grade, but the two gentlemen on the left side were scientists that were given the Nobel Prize in Science in 1953 for discovering the shape of a DNA molecule. The shape is called a double helix, and it simply means that it's a ladder that twists, that rotates as it goes up. Now, the lady on the right side was also a scientist. The two gentlemen, Watson and Crick, had to use her X-ray crystallography technique that she had really developed in order to make their discovery. She was not given the Nobel Prize with them. She passed away of cancer in I believe in her 30s, if I'm not mistaken. But there's no question that her role in their discovery of the DNA double helix was crucial. All right, so let's talk about what DNA is made of. DNA is made of repeating parts called nucleotides. And when you first look at a diagram of DNA, like the one on the screen right now, you might be a little intimidated. You might be like, what is all that? But if you look at it carefully, you realize it's extremely repetitive. Nucleotides have 3 parts to them. So there's a nitrogen base, the A, T, C, or G. And look in the middle of the diagram. You can see A's and T's and C's and G's. There's always a phosphate molecule. Those are the purple circles on the diagram that are running up and down the backbones. And then there's also one deoxyribose sugar. The deoxyribose sugar is a pentagon. It's 5-sided, and it's sort of that salmon or orange color on the diagram. So one nucleotide is a purple circle, a salmon pentagon, and then one of the letters A, T, C, or G. If you look at that backbone, the 2 posts of the ladder, it's super repetitive. It always goes phosphate, sugar, phosphate, sugar, phosphate, sugar, going up. And on the other side, it's doing the same thing, phosphate, sugar, all the way up. The only part that's changing is the middle, and it's not changing that much. A is always across from T. That's the nitrogen base is called adenine, and the other one is thymine, and G is always across from C. So the part that makes humans unique, or a tree unique from a human, is the order of the letters in the middle. And most human beings share with each other something like 99.9% of all the same letters in the same places. There's just a few changes that make us unique, that make us either male and female, or make us lighter skin, or have more melanin in our skin. Just a few changes in our DNA that make that. All right, so there's lots of different ways to draw a DNA molecule, but a lot of them require an art degree. So early on, scientists figured out, hey, there's a much easier way for us to talk DNA and to draw DNA. If we all agree we know the backbone is phosphate, sugar, phosphate, sugar, phosphate, sugar, could we just skip drawing the backbone? And if we know the right side is an opposite copy of the left side, could we just show DNA with one side? So here's how scientists write DNA, A, T, T, C, G, T, T, A, G, C. We know that there's phosphate sugars on either side, and then we can write in what the other side would be. So across from the A is a T, across from the T is an A, T, A, C, G, going across all the way down. Type that in right now and take a moment to see if you can figure out, remember what the opposite letters are. Okay, so now we're going to move on to DNA replication. Replication is the idea of copying the DNA so that the cell can divide. If you remember in our last unit on cell division, we said that replication takes place during interphase. The cell can't divide if the DNA hasn't been copied, otherwise the cell would run out of DNA. So very straightforward, I'm going to give you some steps and then I'm going to make it a little bit more complicated. The first thing that happens is DNA untwists and unzips. Look at the top of the diagram, that's the original parent DNA. There's an enzyme that they're not showing you called helicase that untwists and unzips that molecule. Then there are another couple of or set of enzymes that are called DNA polymerases. And the DNA polymerases travel down the separated strands and they read a G, and so they grab a C. And they read a T and they grab an A and they read. So that's attaching, that's what step number 2 is, attaching free nucleotides to the complementary base pairs on each side. Yes, you have free A's, T's, C's, and G's floating around in your nucleus so that the enzyme can grab them and attach them, grab them and attach them, grab them and attach them. In the end, I want you to really look at the diagram. Do you see that the left side and the right side have been copied and they are now identical? Look at where the yellow ribbon starts. G across from C, G across from C, C across from G, C across from G, T across from A, T. They're identical. What you have created in essence are the sister chromatids of a bivalent chromosome. Now that cell can go through mitosis. One side of the DNA can go into one cell and the other side of the DNA can go into the other cell. Okay, and that's what we're saying here. Notice that the two copies at the end of replication are identical. They are the sister chromatids of a bivalent chromosome. Pause me if you need to write anything in. This is another diagram showing the same thing. If you need to, blow up your screen so you can read the details. But now I'm going to give you a little bit more complicated information than just what this diagram indicates. This drawing down below shows you more detail, and this is where I really want us to be. So it says replication is, of course, a little bit more complicated than I made it sound at first. The two separated strands of DNA, so if you look at the far right side of the diagram, that's the original parent DNA. And there's the enzyme, they show it this time, it looks like a little egg, is untwisting and unzipping. And so the separated DNA strands are called leading and lagging. Find those labels on the diagram. They run in opposite directions. And I don't have time to explain what that means, that they run in opposite directions, but just imagine that the leading strand is like a one-way road and it's traveling one direction, and the lagging strand is a different one-way road running the opposite direction. Well, it turns out, look for the little blue eggs on the diagram that are marked DNA polymerase. DNA polymerases can only travel one direction, adding their nucleotides. So on the leading strand, that works perfectly because they're traveling the direction that the leading strand travels. So it says here the leading strand runs the correct way. So DNA polymerase moves across it swiftly, copying the DNA continuously, nearly as fast as the helicase is untwisting it. This is a movie. It's happening really, really fast. The problem comes in with the lagging strand. The lagging strand is running the opposite direction. So DNA polymerase has to hop on where the two strands are separating. That's called the replication fork. DNA polymerase starts at the replication fork and works backwards. And then when it reaches the end of the DNA, it falls off and a new DNA polymerase attaches at the new replication fork. Because in the meantime, helicase has been traveling the opposite direction, unwinding more and more of the DNA. So each time that helicase moves further, a new DNA polymerase has to hop in and copy that new section. Sometimes people refer to that as backstitching. So the new DNA polymerase copies until it reaches the previous section, then it falls off and the process repeats. I'm showing you with an arrow how each DNA polymerase copies just a little section and then falls off. The lagging strand is therefore formed what we call discontinuously. It's a series of segments called Okazaki fragments that are then joined together by the enzyme called ligase. And finally, I got to say this quick, the DNA polymerase only makes about one error in every 10,000. There are proofreading enzymes that fix it down to 1 per billion, but don't forget that DNA can also be damaged by things like carcinogens and that kind of thing. All right, gotta go. Have a great day, everybody.